Hello everyone! The third qualitative reaction from the Biobranches Biochemistry box will be the one which tests the presence of lipids in our food, particularly butter. We have a box that contains 3 biochemical experiments, and we will use reagents required for the third one. So let's take a look inside the Biobranches box, which contains bottle, gloves, spoons, petri dish, and reagents: sodium hydroxide and denatured ethanol. Today we will make an experiment with a special lipid type called triglycerides. Triglycerides are the main constituents of fat in human and other vertebrates' bodies, as well as in vegetable oil. In simple words, they consist of a head, glycerol, bound to 3 tails, fatty acid molecules. Triglycerides are the primary form of energy storage in our body. A qualitative reaction we want to talk about has an important practical role in human life and is called saponification. It happens between triglycerides and alkali, for example sodium hydroxide, in the presence of alcohol when it's heated. This reaction results in disruption of the glycerol head of the lipid from the tail, which are presented by fatty acids. These fatty acids tails react with alkali and become fatty acid salts, which are known as soap. We are going to start a saponification reaction using butter and sodium hydroxide, and gloves are obligatory here, as we are working with alkali. We will show you the experiment only with butter. However, you can try it on other products such as egg or starch. Put several grams of solid butter into the provided dish. Then add 20 milliliters of ethanol. And then 20 milliliters of sodium hydroxide. Mix it well. Cover, but do not close the bottle completely in order to allow the evaporation of liquid. Next, transfer the thermostatic bottle into the microwave and set it up on the lowest power for 3 minutes. Do not leave the microwave and monitor the reaction to avoid the active boiling and leaking of the solution. As the bottle is hot, use protective glove to take it out. After boiling started, you can see soap foam which emerge as a result of hydrolysis of lipids. It results in the formation of glycerol and sodium stearate. This reaction is called saponification. It is very important for us as sodium stearate has both hydrophobic and hydrophilic parts, which actively removes dirt from our clothes as skin. So now you know how to detect different biomicromolecules, namely polysaccharides, proteins and lipids, in food around, using qualitative reactions. We hope you enjoy the provided experiments.